Good afternoon, everyone. It's Dr. Gershon, and I am absolutely thrilled to be here again for another BetterHealth weekly webinar about healthcare innovation and technology, highlighting truly incredible CEO, entrepreneur founders. And I have an amazing CEO founder here today in Dr. Adam Watson. And he is the CEO of MeCo Diagnostics. It is really a truly mission-driven company that is using novel biomarker technology to approach problems with breast cancer. And then in addition to that, he's going to tell you about the therapeutic component where he's using technology to repurpose generic drugs to find very cost-effective solutions to delivering therapeutic care for breast cancer patients, and as a platform for other disease categories, which he will tell you about. Before I introduce you to Adam, I just want to say thank you, everyone, for just being an incredible audience. The questions that we get, the enthusiasm, it is creating a tremendous open source healthcare ecosystem where companies and CEOs and entrepreneurs are collaborating with industry, with investors. There is this feeling that we can really make a difference and reshape healthcare together in a collaborative effort. And I know I spoke with Adam about this today and in prior conversations. And it's very much a part of the mindset of collaboration. And that's one of the things that I think we want to talk about today, which is, where did Adam's journey begin? Why is he doing what he's doing? And potentially a call out or shout out to people and companies to help in this journey and collaborate. So thank you, everyone, for joining in. And now I want to introduce Dr. Adam Watson, who is the CEO of MeCo Diagnostics. And Adam, it's a real pleasure to be here. I want to start off by perhaps asking you to talk to the audience here about who you are, how you got to where you are, some of the experiences as an entrepreneur. And I know that there are some fascinating things that have just happened over the recent past. And this way we can kind of share a little bit about, the platform and the foundation of the journey, and then take it into kind of where you are as a company right now.
Certainly. Well, yeah, it's great to be here. Thank you, Dr. Gershon. So my name is Adam Watson. I'm actually Canadian. I'm from Canada, Vancouver, BC. I came to the United States to do my PhD at the University of Arizona. And actually, while I was there studying cancer biology, I invented the technology that I am now commercializing. So our company, MeCo Diagnostics, is a tech transfer from the University of Arizona. My co-founder and chief scientific officer was a PhD advisor. So I'm now his boss, which I like to remind him of every now and then. And basically, we never really struck out to start a business. Neither one of us were really had that business focus or business acumen. In fact, it was very late into our journey of understanding the basic science of what we're doing that it became really obvious and imperative that we actually work to commercialize this, because the technology wasn't at a stage that the university could license it to an outside party. It still needed work to be done, and it became clear to me at some point that the only person that was going to do this was me. So I dramatically changed my career focus from wanting to stay in academia. I've always wanted to be a lifelong academic. Quite frankly, I still do. The project and the mission of what we're doing really demanded that I switch focus and I take the mantle of what we're doing and do the studies that need to be done in order for us to get the level of validation of our technology so that we could really bring it to the clinic. So that's essentially how I pivoted from academia, to business. But really at heart, I still am, a scientist, and I do love being at the bench, primarily working with microscopes And so that's how my journey started. And now I wouldn't have ever, expected a few years ago that, I would be, raising venture funds like we have, and talking to oncologists about changing the standard of care for breast cancer. But really, that's where we're at now. And with the clinical validation that we just got, we really are at that stage of, things are moving very quickly. We have a great trajectory, and it's really quite exciting how far we've come since incorporating just over a year ago.
Amazing. Yeah, and maybe we can amplify on that personal side of the journey a little bit before we get into some of the, the science aspects, and what you envision. The people that are around you, your team, and the collaboration partnerships that you have, um, in Southern California. Perhaps you can tell young entrepreneurs a little bit about that personal experience in team building. And sometimes, science can be, quite siloed. You're working in the lab at 1 o'clock in the morning, running your experiments, and now you are a CEO and you're team building. So there's a bit of a different mindset there at times, I would imagine.
Yeah, no, definitely. And again, I has learning as I was going over the last year, which can be, chaotic and frightening. But yeah, so early on, I realized that one thing that I was missing was, I had a very strong technical background, but I didn't have a strong business background. So I knew that when we started our company, we started in Tucson, Arizona, because that's where we were both living as I was finishing up my PhD, working to get our paper published. My dissertation work got published in Cell Reports. And because it's a high impact journal, it takes over a year. Frankly, it took almost 2 years to get actually published from when we submitted. So during that time, we thought about, okay, let's try to stand up this company. Let's apply to get some government grants, like SBIR grants, to get non-dilutive funding. And so, during that time, it became clear to me that starting a company, biotech, medtech startup in Tucson is very difficult. A lot of investors just kind of don't take Arizona startups very seriously. And so I realized, I should move to the coast, essentially, come to San Diego. In addition to a great climate, there's really a hotbed of, tech and biotech here, with a lot of the companies, like Qualcomm and Dexcom, really big name companies that are helping, startups that are, sort of tech focused or, tech adjacent. And so, when I came here, I worked for another cancer biology startup at an incubator called J Labs, which is very famous. It's run by Johnson & Johnson. I really kind of caught that bug, the entrepreneurial bug. So despite working for another company, I was there for about 6 months. I realized I really need to focus all my energy on my own thing that had been kind of in the background for a while. And so I realized that being in an incubator is actually an incredible experience. So we applied to be in EvoNexus, which is a Southern California incubator. And it's been a great experience. It was the due diligence process to get into EvoNexus took 3 months almost. It's very selective, but once we were in, we had a lot of the support that we needed. And, I got a team of 4 advisors and mentors, all domain experts. So either, retired pathologists or very seasoned business executives who, helped raise billions of dollars. They were then on our team, right? And so I could sort of call upon them, when I needed some advice or get some connections. And being in an incubator was transformative for us, because as someone with very little business experience, and my co-founder, he's a professor, he doesn't have any business experience, we were still in the very early stages. And in order for us to have that sort of outside, credibility, external credibility, getting into a selective incubator, and there's a few good ones out there, that it really can help launch a company. And as for example, we're a perfect example. We got venture funding, about $500,000 in venture funding, which was absolutely transformative for us and helped us get to the big milestone that we just got to a couple weeks ago. But getting that venture funding, the first big check, as everyone will know, is difficult. We got it less than 30 days after getting admitted into the incubator. And, I asked our funders, point blank, let's be candid, would you have funded us if we were not in EvoNexus? And they said no. And, part of that reason is they know that they've previously invested in EvoNexus portfolio companies. that have done well. So they use that as a sort of a proxy of legitimacy and also confidence in who's supporting us on a day-to-day basis. So I think it's absolutely for people out there that are like academics. Maybe they're postdocs and they're working on something that they know has commercial value, but they don't really know how to spin out. Sure, there's lots of resources out there, and for the best resources initially are going to be at whatever institution you're at, because now every school has sort of a tech transfer office that will help you do this for free, and you should take advantage of those resources. But then after that, after you have sort of the core team and you have a working sort of pitch deck, you have to have a real plan that you can defend. At that stage, I think it's a really good idea to go an incubator if your team is lacking this sort of startup experience, specific startup experience that you'll really need for the first few years, or at least the sort of pre-seed seed stage. And so I would absolutely recommend looking into incubators. At the same time, to be a bit careful, I will say that I won't name any names, but there are some accelerators out there, particularly ones that no longer exist as of very, very recently. But I won't say any names. But sometimes you'll get contacted by accelerators, and they'll say they won't totally tell you the structure of the deal, but they'll say, hey, do you want to apply to come in? And you apply, and they say, you're admitted, you're fantastic, we're excited. And then they kind of change it's a bait and switch. And then just before they say, oh, by the way, you do have to spend 10 grand because we have to pay our mentors. And so I actually did decline that opportunity. And so I would say that as exciting as it is to get some of that external validation early on, or people that seem to care what you're doing, there are some, I would say, bad actors out there in the accelerator world, and there's some great actors. So be careful with who you're going to give money to, especially when there's very little money early on in terms of getting help. There's a lot of free resources out there, and it's definitely worth finding those free resources to help you.
Terrific. Well, let's shift over first to thank everybody for dialing in today. Of course, I want to thank Jenny Fernandez, who is with us all the time, and Dr. Joshi, who is the CEO of Clinical Trial Hero, just a tremendous company. I want to thank Vitaly, who I'm not sure where you're dialing in from, Vitaly. I see someone asked you that, but welcome. And I want to now talk a little bit about the science, because that is your background. As you said, you are an academic, and you have created this new technology. Tell us about the technology and how that is potentially going to be a game changer for patients.
Yeah. So as I said, we didn't start out with dreams of commercialization. Really, I spent 6.5 years just trying to understand what's called the mechanical microenvironment of cancer cells and tumors. And in simple terms, it's the physicality of a tumor, and particularly breast cancer. And this is actually something that everyone is familiar with. In fact, the oldest observation in oncology is a stiff lump in the breast that then grows and becomes problematic, and then spreads throughout the body and kills the patient. We've known about this for literally 2,000 years. And so for a long time, people thought that the change in the physicality of a tumor, i.e. getting stiffer or getting inflamed, getting red, like the actual physical changes, we thought that these were simply consequences of disease progression. And in the last 20 years, we've really begun to realize that physical changes, especially stiffness, tumor fibrosis, it actually can cause certain disease phenotypes, disease behaviors. And so part of what we published as part of my dissertation work was showing that tumor fibrosis, which is essentially an accumulation of sort of collagen and hyaluronan, these things that sometimes we want more of, like in our face, to look younger. But I'm sure everyone is aware of the effects of fibrosis on the outside of your body. If you get a deep cut, and it heals when you get a thick raised scar that's a little bit firmer. So that's what fibrosis is, and we're all sort of familiar with that. When it happens in breast cancer, it's what leads to the lump that you can feel. And really the issue for us was to understand how this can be problematic for patients. And so really that's what we spent a long time understanding the mechanism of how exactly cancer cells respond to changes in stiffness, and then become really bad actors and then spread throughout the body and cause that mayhem. So really what I invented along with my PhD advisor was what's called a predictive biomarker. And so what this does is allows us to stratify patients into sort of risk categories based on how their cancer cells are responding to physical changes in the tumor, particularly fibrosis and fibrotic stiffness. And so we did a lot of preclinical experiments showing that the same cancer cells, if you put them in fibrotic stiff environments or soft environments, and then they're introduced into animal models, the metastatic ability, like the dangerous capacity of these cells, is fundamentally driven by how they are preconditioned, let's call it, or mechanically conditioned. And so that's actually the name of our company, MeCo Diagnostics. It stands for mechanical conditioning. And this is essentially, mechanical conditioning you can think of as a stiffness response. So how are some cells responding to stiffness and other cells are not? As an analogy, I'm sure everyone out there knows there's people out there that they work out forever and their bodies don't change. Other people can just, pick up some weights or go for a run, and all of a sudden they adapt, their muscles adapt. So they're very well mechanically conditioned. And so just how some people are different in that, and some people can grow muscles quickly and some people can't because of mechanical conditioning, cancer cells are different too. Our technology is able to identify the patients that have cancer cells that are highly mechanically conditioned by fibrosis. And as a result, they're going to have really bad disease progression. And so those are the patients that we can then use anti-fibrotic drugs with. And really the great part of that is anti-fibrotic drugs already exist. There are 2 that are FDA approved. They are oral drugs. They are low toxicity. They're well tolerated. But really the most important feature about anti-fibrotic drugs is that they're becoming generic very, very soon. In fact, the first one just became generic and available last month, this month. It's called pirfenidone. It was formerly a Roche drug. Now it's generic and it's much less expensive. The drug that we are repurposing is called nintedanib. And it's a Boehringer Ingelheim drug, and it's currently used for fibrotic diseases of the lung, so non-cancer diseases. But it costs about $12,000 a month right now. It's a targeted therapy. But in 9 months, it's going to be generic. And then in 2025, there's going to be 2 generics on the market. So the cost is really going to come down. And the reason why this is important is that at the same time that our company is clinically validating a technology to predict which patients will benefit from anti-fibrotic drugs, particularly this one that's becoming generic in 9 months, the cost is going to come down. And the reason why that's really important to me is, coincidentally, when I was in grad school inventing this technology, my aunt was diagnosed with advanced cancer. And despite the fact that she had excellent health insurance, and despite the fact that she formerly worked as an executive at Cedars-Sinai, which is where all the celebrities go to get cancer treatment, in Hollywood, very, very, great, fantastic place, for research and treatment. Despite those 2 features, great insurance, great, network, she ended up spending hundreds of thousands of dollars in out-of-pocket expenses for non-reimbursable treatments. And the sad fact is, I didn't realize this, a lot of people don't realize this, but even a Cadillac gold plate insurance plan that you have, it is not the shield you expect it to be when you need it the most. And the reason for that is there are a lot of options out there for treatment, and not all of them are going to be covered by your insurance plan. And so when all hope is seemingly lost with standard of care treatments, the treatments that are covered by your insurance plan are reimbursed, your disease keeps progressing. You're going to do everything you can for yourself or your loved ones, right? And for my aunt, that meant remortgaging her house, draining her daughter's college fund to pay for immunotherapies that were not covered then. And she still died, right? And so she left her family's debt, and it really showed me, wow, this is a big problem. Cancer is not only a health problem, it's also an economic problem, right? And the problem with new drugs is that we're getting better and better at treating cancer, and hopefully some will be curative soon for some of the major cancers. But the thing is, you can't actually solve cancer as a holistic problem with new drugs because they're so expensive. What you're doing is you're transforming a health problem into an economic problem. And that's really not what we ought to be doing. So I don't, I don't want to hate on big pharma for developing new drugs. We need new drug development, don't get me wrong. But what we also need is old drug repurposing. And so that's what we're focused on, is repurposing drugs that are becoming generic, they're becoming cheap, so that we can address the sort of economic crisis and catastrophe that a cancer diagnosis, can give to patients, because, medical bankruptcies are on the rise, cancer incidence is on the rise. So I think we do need to sort of, really come at this all hands on deck and look at all the possible treatments that we can come up with, both, new treatments and repurposing old treatments to sort of address both the health and economic problem of cancer. So that's really why we are, our mission is to repurpose generic drugs, inexpensive drugs, used for other diseases, for breast cancer first, and then prostate cancer after. So that's really what fundamentally differentiates us, I think, from most other startups that are looking to tackle cancer.
Terrific. I mean, combining, a biomarker with a therapeutic, it makes perfect sense. What I would like to explore a little bit is a little bit deeper dive on your technology and a little bit about the research and the invention. Now, I understand that there is a correlation between the fibrotic density or level. There must be some scoring mechanism. Maybe you can expand upon that. First, the analysis of the fibrotic microenvironment. And by the way, in terms of microenvironments, just tremendous advances in understanding about the differences in these environments, the different cells within these environments that react, don't react, or partially react to therapy. So, really what you're doing, I think, is so additive to many other things that are happening. That synergy, again, speaks to, bringing to a public forum companies like yours, understanding the technology, and then hopefully light bulbs go off in people's heads, and they're like, you know what, I have to contact Dr. Watson because I've got an idea that might fit. So that is my way of a shout out to everybody who's listening. On behalf of MeCo, if you have ideas, please contact me or Dr. Watson. This is about an open source platform for sharing and collaborating to better healthcare. So let's talk a little bit about the science, that a little bit of a deeper dive in understanding the analysis that you're doing about the fibrotic tissue, and then the correlation of that fibrotic analysis with the effectiveness of essentially off-label use drugs. So you have a drug that's used for fibrotic disease in the lung. How do you know that that's going to be effective for the anti-fibrotic effect for non-lung tissue?
Yeah, so another thing to point out that's really quite interesting, there's a couple things that are fundamentally unique about what we're doing. But one thing is, we're using this anti-fibrotic drug, to treat breast cancer as a disease. What's interesting is we're not using the drug to target cancer cells. Now, the genes that the drug targets, it targets 3 kinases. Those kinases are expressed in the cancer cells and they are targeted, but that's actually not why we're using the drug, which sounds a bit weird. What we're actually using the drug for is to target the normal cells in the tumor microenvironment, like particularly fibroblasts. And the reason why we're targeting the fibroblasts are the cells that make collagen. They make connective tissue. And so fibroblasts are the cells that get tricked by the cancer cells into making more collagen. And then this makes the environment fibrotic and stiff. And then the cells respond to that stiffness and start to behave really badly. So we're interrupting that cycle in the tumor microenvironment by preventing the fibroblasts from going crazy and making too much collagen. And so what happens is anti-fibrotics, they start to soften the tumor microenvironment, and then the cancer cells stop to behave badly. And so we're not actually aiming to kill the cancer cells with these drugs. We're actually just changing the tumor microenvironment to make the cancer cells less aggressive and then more susceptible to traditional therapies. And so I should mention that we are not aiming to displace any therapy. In fact, our clinical trial that we already have run, which we just finished up with the government of Spain at the Spanish National Cancer Center, and we can talk a bit about that, but also the clinical trial that we're starting next year at George Washington Cancer Center. In both cases, the control group is standard chemotherapy, so what patients would be getting anyways, and then the experimental group is chemotherapy plus anti-fibrotic therapy. And so one of the ways that it's thought that this is actually helping to work is that by softening the environment with the antifibrotics, the chemotherapy can actually do a better job of killing the cells for two reasons. One, the cancer cells have less of a defense because they're less aggressive. But two, soften the environment, it actually changes what's called the partial pressure. And actually this allows diffusion of the chemotherapy into the tumor, because if the chemotherapy can't diffuse into the center of the tumor, it can't kill the cancer cells. So I do want to say that it's not something, it is additive. That's a perfect word that you said. We're not seeking to replace anything. But in terms of the actual science, so what we did was, we started with a model cell line, and we looked for a cell line that had particular properties. And those properties are, when you grow these cells, cancer cells, they're called SUM159 breast cancer cells. When you grow them in a fibrotic environment, they become very aggressive and very metastatic in animal models. When you take those same cells and you just grow them in a soft environment that mimics healthy normal tissue, they lose their invasive properties, they lose their aggressive properties, and most importantly, they lose their metastatic properties. They don't metastasize, in animals. So we knew that this was a perfect model for, stiffness being aggressive and softness, normal tissue softness being preventative. And so then what we did is we did RNA sequencing on both the stiff conditioned cells and the soft conditioned cells. And so RNA-seq, we did whole transcriptome, which means we looked at all 20,000 genes that are being expressed and looked at the levels of each of those genes. And then we simply compare the differences in gene expression across all genes in both the stiff conditioned cells and the soft conditioned cells. And so using that, we were able to derive a signature, or we call it a score. And so our technology is called the MECO score, or mechanical conditioning score. And so the higher the score is, the more like they are the cells that we know are very, very aggressive in a stiff environment. And then a low MECO score, those are cells that are really non-reactive to the stiff environment. They're non-aggressive, they're going to be non-metastatic. And so when we apply that score to patient biopsies that are taken for the diagnosis of cancer, right? So we actually don't need to take any additional tissue. This is a non-invasive test. The tissue sample already exists because it was taken out as a biopsy and given to a pathologist, and the pathologist looked at it and said, yes, this is breast cancer, you have breast cancer. So we can take some of that tissue that already exists, run RNA sequencing on it, and then look at the genes in our score, and there's 1,004 of them, and then using that measurement of those 1,004 genes, we can then assign a MECO score. And so the higher the MECO score, we know that those cells in that patient's tumor are very aggressive and they are likely to metastasize. But also we know that those are the cancer cells that ought to be targeted using anti-fibrotic therapy. So that's how we know which patients really ought to get anti-fibrotic therapy, because they have a high mechanical conditioning score. And so this is really what completely differentiated us from previous attempts to use antifibrotics in cancer, because people have tried before, but they were always using the presence of fibrosis as the determining factor of whether or not to use antifibrotics. And that sounds logical, right? You should use antifibrotics when there's fibrosis. It turns out that doesn't work, because, and this is something that everyone listening right now actually already understands intuitively without knowing it, is that most lumps in the breast are benign, as we know, thankfully, but they're fibrotic. So the presence of fibrosis isn't sufficient to be dangerous. So you can't use the presence of fibrosis to determine if someone should have anti-fibrotics, as obvious as that might seem. You need to actually understand whether the cancer cells in that tumor are responding to the fibrosis in a bad way, and that's what the MECO score tells us. And so then we can actually just basically filter out the patients who aren't benefit from antifibrotics, even if they have very fibrotic tumors, they're just not going to benefit, and then just be able to identify and treat the patients who really will benefit, and they definitely need extra help because we know they have very aggressive tumors. So that's essentially kind of a broad overview how our study.
So if I understand, there is really a causative effect here. So the fibrosis is triggered in cancer. The fibrosis then is some type of catalyst or activating agent for certain cancer cells, which then respond to that microenvironment and become aggressive and potentially more likely metastatic.
Exactly.
Now, so very, very interesting. So then I guess the logical next step is, when can you diagnose with this biomarker, what is the earliest stage that you can diagnose for the use of these anti-fibrotics to be effective? Because it would seem intuitive that if you capture this very early on when there's trace elements of fibrosis, that it would prevent that progression. But at the same time, there is this combination or sequence of things that isn't necessarily intuitive. As you were saying, benign lesions can be fibrotic, but there's some other agent in there that transforms from a benign to a malignant. So maybe you could talk a little bit about that, because that's very interesting. Like, how early can you detect? And then, is your score sensitive at different stages in an equal sensitivity and specificity? How are they going to use this?
Yeah, so and it's a good question, and it brings up a really interesting point, and in perhaps some ways an unfortunate point. One of the ways that anti-fibrotics have been trialed in breast cancer in the United States, and in a way that failed, unfortunately. Now, I knew that it was going to fail, but it was because of our understanding of the mechanism. But you have to use anti-fibrotics as early as possible, and before cancer cells have spread throughout the body. Now, once cancer cells are metastatic, it no longer makes sense to use anti-fibrotic therapy because they've already left the primary microenvironment, which you're actually treating with anti-fibrotics. So unfortunately for metastatic patients, it's too late. So really, and the unfortunate thing is, I think anti-fibrotics got a little bit of a bad rap because they were trialed in metastatic patients and it didn't work. So people thought, oh, anti-fibrotics don't work for cancer. They were just using the wrong patient population. So really, the earlier the better. And really, we see our technology as being, consolidated with all the other predictive biomarkers that exist. And I'll give you an example. Many people know that breast cancer is treated with a lot of different modalities of therapy, right? There's chemotherapy, there's hormone therapy, which is endocrine therapy. There's radiation therapy, there's immunotherapy. Now there's cell cycle inhibitors. So there's really a lot of, and then surgery, of course. And so there's so many different modalities of treatment. There are now predictive biomarker tests for all of those different modalities of treatment. And it's a little bit unfortunate that we are taking 6 different pieces of tissue, which hopefully there's enough to do that, to do 6 different tests, to tell a doctor if patients should get 6 different modalities, which of those, to really have personalized care. So what we're hoping that will happen, and essentially this is part of our business development plan, really is to partner with a company that's already doing sequencing of tumors, early stage tumors, right after diagnosis, to know as early as possible what is the best treatment path. So with the same piece of tissue, you should just frankly be able to, we can already theoretically do this. You do one sequencing reaction, right? You look at all 20,000, and then you simply apply different algorithms to that sequencing data to get different answers about what kind of treatment you should have. So this is very, very efficient, right? It saves cost, right? Running an algorithm doesn't cost any money. So essentially it's like added value to, something you've already done, you spent money on to do some sequencing that maybe costs, $500. Now you can get all these different answers. So it's going to help bring down the cost of care overall, because in addition to, leveraging generic drugs that are cheap, our test can be built on another test, sort of piggybacked on another test, so that we can help, hopefully, bring these costs down in the long term, so that the cost of the testing is cheaper and the cost of the therapy is cheaper. So really, we actually do hope to integrate our testing at the same time as other predictive biomarker testing that's happening, which happens as early as possible. Really, upon diagnosis, it's almost like reflexive now for patients to get certain testing. Now, there's certain subtypes of cancer, like I'm sure people have heard of HER2 positive cancer. Now, it's a very active cancer. And because we have very effective drugs that treat HER2 positive cancer, it's a reflexive test. So as soon as you have breast cancer, they're immediately going to see if you're HER2 positive. And if you are, you get HER2 targeted drugs. So what we hope is that the MeCo score becomes a reflexive test as well. So as soon as you have early stage breast cancer, especially one that is considered to be high risk based on, classic pathologic factors, like the grade of the tumor or the Ki-67, there's a bunch of ways that pathologists can determine or estimate, the risk to the patient just on classic pathologic characteristics. So patients that are known to be high risk already, I think reflexively they should get a MECO score test to know if anti-fibrotic therapy is going to be indicated. And if so, it should be used concurrently with chemotherapy, because that's actually what we're doing in our clinical trials, is using it in combination with chemotherapy or hormone therapy. And that's also something we're going to be exploring in our upcoming clinical trials.
It's so interesting because all of this is additive. The work that you're doing is synergistic with a lot of other work that's happening, as we talked about earlier, in microenvironment therapies, but also in AI and pathology, and being able to use AI with a higher predictive value in really identifying cancers earlier and earlier, I think is also, a very exciting synergy between what you're trying to do, because you're talking about potentially, I would assume, even, I don't know, this is really a question to you, breast cancer in situ, is that something that you would also be able to detect, or is that too early?
So our test won't detect cancer. We're not a diagnostic in terms of diagnosing,
the fibrotic tissue.
Yeah, so it really comes after in sequence. Necessarily, it comes after in sequence from the diagnosis of cancer by the pathologist. After that happens, then it triggers these other tests that are becoming sort of reflexive, as I say. So, right. As soon as we know you have cancer, well, we know, should you take chemotherapy, right? That happens for basically every patient now. Should this patient get chemotherapy or not? And one of the reasons for that is we've been doing a lot of over-treatment of patients for a long time with chemotherapy, because the thing is, 70% of women with early-stage breast cancer, pre-metastatic cancer, 7% of those patients will never become metastatic. They'll die of something else in their life, and that's fantastic. So as a result, we've learned that we don't need to necessarily, throw every single treatment at a patient, because chemotherapy are detrimental to patients in a lot of ways. Radiation is detrimental. I mean, you can treat cancer with radiation and cause a new cancer with radiation, right? I mean, it's kind of butchery if you think about it, and we will think about that 50 years from now. But so, but they still work. And so what we're doing now, and this is, again, AI is helping us do this as well, and not us personally, I mean, as a community, is that it's helping to understand, how can we limit the use of, toxic therapies like chemotherapy and radiation, and really save them for patients who really need it. And so there's this confluence of better understanding of prediction of therapy response. And so again, the MECO score and anti-fibrotic therapy is additive to our growing understanding of this sort of comprehensive understanding we have of how a patient ought to be treated to limit this sort of toxic side effects, if they don't necessarily need it. And everyone's waiting for sort of, people talk, very superficially about a cure for cancer. I mean, certain cancers already are functionally cured, like CML, testicular cancer, very, very high cure rate, over 95%. With breast cancer, we're getting there, we're creeping up. But I don't think that it's actually theoretically even necessary that we have a game changer drug now, because if we simply catch all breast cancers earlier, right, with improved screening methods, lower cost screening methods, lower cost mammograms, and then of course AI-enabled mammograms, because there's not enough pathologists out there to score a mammogram, and you need multiple pathologists to sort of agree, and then patients that there's false positives or false negatives, and then this of course leads to over-treatment or under-treatment. So the more that we actually get better at early detection, which is not what we do, we don't detect cancer, but when other companies are getting better at early detection, that helps us get in there faster to then assess the patient with a MECO score to know if they should get anti-fibrotic therapy. And then all these other companies working on their predictive biomarkers to know, should a patient really get radiation at that point, or hormone therapy or immunotherapy. The earlier we do all this, and the fewer patients that kind of fall through that net, then the more patients really are going to be functionally cured. And so I think it's very, very reasonable to expect that in the near future, we will have a 95% cure rate of breast cancer, because we're simply just catching them really early. But of course, to get there, that's not just a scientific problem, right? These are socioeconomic barriers, there's political barriers, there's regional barriers. And so, at least for us, one of the ways that we are helping to, give patients access to anti-fibrotic therapy is that we chose a methodology, RNA sequencing, in which case every patient in the United States, no matter how remote they are able to actually participate in testing. And I'll give you an example of why sometimes this doesn't work. If you made a predictive biomarker test using PET imaging, right, using a PET scanning machine, $2 million machine in a special room in a hospital, generally in an academic medical center, it might work great. You might use this PET machine to make any sort of great prediction, and that's going to help the patients who live near the hospital that has the PET machine that can have access to it. It's not going to help people in rural areas. So the MECo score, because it's done off of a little piece of tissue on a little slide that you can ship to our facility, and that we or sequencing partner's facility, you can be a patient anywhere in the United States, frankly anywhere in the world, and get a MECo score done, and then have a generic drug prescribed if it's appropriate. And so that's another way that I think everyone in the community sort of needs to work together to come up with solutions, not just for people in major metropolitan areas, but really a way that patients everywhere can have access, to the newest and best therapies.
Just absolutely fantastic. The speed in which you've done all this is remarkable, and I can't wait for you to come back in another webinar or a podcast, which we're going to be starting in the fall, to give us updates. I know that there are going to be tons of questions. For those people that do have questions, please feel free to email me and I will pass them on to Dr. Watson. I want to thank everyone again for their participation here. We've got some exciting comments from Chad, who has chimed in, excited to see where the technology takes us in these early cancer diagnoses. We've got Swati, who is very involved with cancer in her company, involved with MD Anderson down in Texas. We've got Phil, and of course, Jenny and Dr. Joshi. I really thank everyone so much for tuning in, taking part in this community sharing of incredible CEOs, incredible entrepreneurs who are really reshaping healthcare. And if you are involved in a company that you think would like to come on the webinar and speak to the community, please feel free to contact me. It's a very exciting time in medicine and in science. I am absolutely thrilled about talking about a cure rate potentially at some point, or certainly a treatment rate that approaches greater than 90% effectiveness for breast cancer is absolutely, I think, insights. And I agree with you that over the next several years, these technologies are just absolutely going to change healthcare for everybody. And the key piece of this, I think, that you highlighted several times, which is access. And so that's really a very important part of what we're looking at for a number of companies, is this diversity gap, whether it is travel distances, which you pointed out, or socioeconomic issues. McKinsey has done a study which looked at social determinants of health and their correlation to the diversity gap. And, different diseases certainly manifest smaller or larger gaps. It'll be very, very exciting to be able to roll out your therapy over the next few years as an additive mechanism to close that gap, because these are affordable tests, affordable treatments. So I can't thank you enough for being on today. It's, limitless questions I have in my head, but we're a little bit short on time today. So please come back and please tell everybody about the incredible things that you're going to be doing over the next several months and several years. It's exciting to host you today.
Well, I think I really appreciate the opportunity, and I'll never turn down an opportunity to share our mission with people, because I think there's really so much opportunity for synergy out there, and we're moving quickly, and we need a lot of help to get where we want to go. So I appreciate the opportunity.
Absolutely fantastic. Enjoy the rest of the day, everybody. Thank you for tuning in. Thank you, Adam. Stay on the line for a minute, and I'm just going to say goodbye to everybody today. And please tune in next week. Next Tuesday is July 4th, but we're going to be hosting a company on July 5th, which I think you're going to find incredibly exciting called Pepper Bio, doing some really outstanding things in healthcare. So please tune in on the 5th at noon. Have a wonderful rest of the afternoon, everyone on the East Coast, and a wonderful day for those people elsewhere in the world. Bye-bye.